Hello everyone, welcome to Dentistionary. We are back with human physiology lecture and today we are going to begin a new unit called Special Senses where we'll teach you some selective topics and our topic for today's lecture will be on visual process and its pathway. So without wasting much time, let us begin our today's lecture. So that's a reminder. You can ask your doubts in our comment box. We also provided the PDFs of the slides in the description box. So let's begin. First, we have the wall of the eyeball. The wall of the eyeball mainly comprises of three main layers, the outer layer, middle layer, and inner layer. The outer layer is also known as tunica externa, middle layer known as tunica media, and the inner layer called tunica interna. The outer layer comprises of cornea and sclera, middle layer choroid, ciliary body, and iris, whereas the inner layer comprises of layers of retina. Please note, layers of retina can be asked as a short note for 2 marks. There are basically 10 layers of retina, namely the layer of pigment epithelium, layer of rods and cone cells, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic cell layer, layer of nerve fibers, and lastly, the internal limiting layer. Please write this name of the layers in the sequence given. Moving to our next slide, we have the rhodopsin cycle. You guys have already read rhodopsin cycle in biochemistry in vitamin A. So, this is just a division for you all. In rhodopsin cycle, the rhodopsin in presence of sunlight or the light energy gets converted into beta-rhodopsin, which further gets converted into lumirhodopsin, meta-rhodopsin 1 and meta-rhodopsin 2. This meta-rhodopsin 2 is the active form of rhodopsin. This divides into two products, namely the scotopsin and all-trans-retinol. All-trans-retinol in presence of dehydrogenase NADH2 converts into all-trans-retinol. Now this two trans form gets converted into cis form, namely 11-cis retinol and 11-cis retinol, which along with scotopsin forms or resynthesize rhodopsin. Hope so rhodopsin cycle is clear to you all. Please mark it as important. Moving to our next slide, we have the differences between rods and cone cells. Obviously, you all know the differences between rods and cone cells because we have read this topic since class 10. In rods and cone cells, the main difference is rod cells helps in dim light vision, whereas the cone cells are responsible for bright light or color vision. Rod cells comprises of rhodopsin pigment and the cone cells comprises of photopsin pigment. Rod cells are more in number, nearly about 120 million, whereas cone cells are 6 to 7 million. Rod cells are predominant in the extrafovea region, whereas the cone cells are concentrated in the fovea region. Okay, let me tell you one thing. If the differences are asked, it will be mostly of 3 marks. So max to max 5 to 6 points are enough to complete your answer. Okay. Moving ahead, we have a disease called night blindness. It occurs due to deficiency of vitamin A in your diet. So what is night blindness? Night blindness is defined as the loss of vision when light in the environment becomes dim. It is otherwise called nyctalopia. Please mark the spelling nyctalopia or defective dim light or scotoptic vision. Causes, as I said, it occurs due to deficiency of vitamin A, which is essential for the functions of rod cells. Deficiency, what are the causes of this deficiency? The diet containing less amount of vitamin A or decreased absorption of vitamin A from the intestine. Clear? Next, we have the field of vision. What is field of vision? The part of the external world that is seen by one eye when it is fixed in one direction is called the field of vision or the visual field of the eye. Suppose this is your eye. So the vision, this part is called your field of vision. Clear? We have the two main types, binocular vision and monocular vision. Binocular vision is seen in human beings and some animals, whereas monocular vision are seen in lizards, dogs, What are binocular vision? These are the type of vision in which both eyes are used together so that a portion of the external world is seen by the eyes together. Please mark it. Here both the eyes view the same thing at the same time. Example in humans and in some animals. The eyeballs are placed in front of the head. Yes, the eyeballs are present in front of the head, whereas in monocular vision, the eyeballs are present at the side of the head, which we will discuss in our next slide. The monocular vision is mostly seen in dogs, rabbits, horses, lizards, and many other species. These are present at the side of the head, so visual field does not overlap so much. Means it overlaps, but to a very small extent. Please mark the difference between monocular and binocular vision. Okay, let's move ahead. We have the division of visual field. Temporal and nasal division and upper and lower fields. Temporal and nasal field division, upper and lower field division. As the name suggests, upper and lower field. The upper part, lower part. Temporal and nasal field, the one which is close to your nose. If suppose this is your nose, the one which is close to it is the nasal part, this yellow portion, and the one which is away from it, this red part, is your temporal portion. Clear? Next, mark this slide as important for your long note as well as for short note. Visual pathway. This diagram you must include in your answer. 90% of your answer will comprise of this diagram. And the remaining 10%, you can write 2 to 3 points, 2 to 3 lines in the points which I will show you now. The optic nerve, the optic chiasma, optic tract, lateral geniculate body, optic radiation, and visual cortex. 2 to 3 points regarding this is enough to complete your answer. Next, we have the applied physiology. Applied physiology comprises anopia and hemianopia. Any injury to any part of the body of the optic pathway causes visual defect, and the nature of this defect mainly depends upon the location and the extent of injury. So, what is anopia? It is the loss of vision in one field. One complete field loss is called anopia, whereas if that vision loss is found in half of the visual field, it's called hemianopia. Clear? A complete loss called anopia and half loss is called hemianopia. There's a chart. Anopia is of two types, right and left anopia, whereas hemianopia is of homonymous hemianopia and heteronymous hemianopia. So what is homonymous hemianopia and heteronymous? Homonymous hemianopia means loss of vision in the same half of both the visual field, whereas heteronymous means loss of vision in opposite half of the visual field. Clear? So that's the end of today's lecture. Next, we'll cover up the accommodation reflex in our next slide. Till then, keep learning and keep showing your love towards dentistry. Thank you.